this is added sometimes instead of phosphate. It's an ADP with the ribose stuff on it, so it's ADP ribosylation. Turns out in the biological warfare that goes on between us and bacteria, this is sometimes used by the bacteria against us. Cholera toxin does this. To one of the human proteins in the lining of the gut, there's a protein there that cholera toxin puts an ADP ribose on it. Very big post-translational modification. This is an important protein in the cell. This is involved in hormone response. And this particular one in the gut controls a transporter that spits out electrolytes into the gut, takes them out of the cells and puts them into the gut. When the toxin does this trick to that transporter, it locks it into the on state forever. Normal hormonal response would shut down the response, but with post-translational modification, this transporter stays on forever. You're pumping out electrolytes, and people die of diarrhea because of that, because the water follows the electrolytes. So this is an example in the world of transport proteins, but it happens in enzymes too. You can put an ADP ribose on other enzymes to fine-tune their activity. Maybe the most general mechanism, though, is this third kind, the allosteric regulation mechanism. Many metabolic enzymes show this kind of thing. And here I've put up a hypothetical metabolic pathway. We're starting with some nutrient A, and you're making various products out of it. It branches, so you can make this amino acid or that amino acid out of it. There's a branch point. How, in theory, would a pathway like this be regulated? A very common way you see is a feedback loop. So two products from one precursor. You've got an enzyme going this way, another enzyme makes that product. You can have feedback loops where this product inhibits that enzyme D allosterically. This end product inhibits that enzyme X allosterically. And allosterically means at another site. Allosteric means other site. So the enzyme binds its substrate here. It's got a binding pocket for some regulatory molecule over there. So the regulators don't have to look like the substrate at all. If the substrate looks like that, the activator or inhibitor can look totally different. So you get feedback inhibition from distant metabolic products that don't look anything like the substrate. Here's a real-life example of amino acid biosynthesis where isoleucine is made through this long pathway. It comes back to inhibit the first step allosterically. One of the most useful properties of allosteric regulation is this. Okay, there's in theory no constraints on this. It doesn't have to be a substrate analog. And many times the regulatory molecule adopts here is something that tells the enzyme about the energy status of the cell. So you can imagine in an energy generating pathway like glycolysis, you don't want to be doing glycolysis all the time. Sometimes you want to do a lot of it, sometimes less of it. And the enzymes have to respond to the energy needs of the cell. If you're really low on ATP, you want to give the go signal to glycolysis. You want to have the glycolytic enzymes ramped up or ramped down in activity by energy molecules binding to them. And that's exactly the sort of thing that happens. Here's the Krebs cycle. We get a ton of ATP out of that. The enzymes in there, many of them are inhibited by ATP. So isocitrate dehydrogenase is just one of the enzymes in the cycle. The cycle makes ATP. If you already have a lot of ATP, it's a feedback inhibitor. It says, slow down, we already got a lot of ATP. It's also got a site where if ADP binds, it's a go signal, it stimulates it. So because of this flexibility, you can design enzymes that when energy is low, that means you got a lot of ADP. It says, let's ramp this up. If you get a very high level of ATP in the cell, it says, okay, we got enough, slow down. So by allosteric regulation, the energy status of the cell controls a lot of metabolic pathways.